Good morning, everybody. It's once again a great pleasure for me from Food Focus Adel Kroch to welcome you to this fifth webinar that we are hosting with the DSA. Jackie and Yompi from the DSA have worked together to ensure that throughout the year that they tackle topics that will definitely add value to everybody within the dairy industry, and this morning it's no different. So we are today going to talk about biofilms today in dairy processing industry, which has got a very big concern in terms of the quality, because it threatens the quality and the safety of dairy products. We are going to look at the growth of biofilms, what's the primary reason for it, and then we will also be discussing what biofilms are, how they are formed, and what we can do, mitigating strategies to prevent them. So we've got Jeff. Thank you, Jeff, from Nutrichem Bayman, that will be introducing our speaker today, who is Justin Peterson. So just quickly about Jeff. Jeff joined Nutrichem in October 2019 as the National Sales Director, and he is been actively selling specialty chemicals to the food and beverage industry since 1990. And he likes to provide the best practices for the best hygiene outcomes for all his customers. Jeff, thank you so much for being with us. I'm going to hand over to you now so that you can introduce Justin, which are in the very early hours of the morning being part of this presentation this morning. So thank you very much. Over to you, Jeff.
Thank you, Adele, and thank you for hosting us on our behalf. On behalf of the South African Nutrichem team, I would like to welcome Justin as our subject matter expert today. Justin, as Adele has just confirmed, has sacrificed some of his beauty sleep. He's presenting all the way from America, and as I understand in America right now, it's just shortly after 2 a.m. in the morning. So Justin, thank you very much. We do appreciate your time and sacrifice to support us, your colleagues here in South Africa. To everyone out there, this topic, biofilm, is a hot topic, forever debatable, and I trust you will enjoy the presentation and the information that Justin shares with you. So with no further ado, I would like to hand you over to Justin. Justin, away you go.
Thanks, Jeff. Good morning, everybody. I'll just dive right into it here. Okay, so first of all, what is a biofilm? Technically, a biofilm is defined as an assemblage of surface-associated microbial cells that's enclosed in an extracellular polymeric substance. It's in a matrix. It's essentially a home. You can think of it more simply as a biofilm is a microscopic city in which microorganisms can live and thrive. Biofilms form because some bacteria excrete a sticky substance. It's predominantly a polysaccharide, and that polysaccharide adheres to environmental surfaces like stainless steel, forming the matrix of that city. A biofilm is a natural habitat for microorganisms. It's built by microorganisms. As we discussed, it's a pretty disgusting picture over there to the right, but that is just a very good visual of what a biofilm can develop into. It consists of a multitude of populations of microorganisms, proteins, fats, sugars, carbohydrates, byproducts of the microorganism's metabolism, acids and mineral scales, polysaccharides can all become encapsulated in that biofilm, in that matrix, polysaccharide matrix. Again, it's a really sticky substance that we've duplicated in the lab, and it's just, if you touch it, it's just really sticky and kind of gross. A biofilm can be formed within 8 to 24 hours after a surface is cleaned, so they form relatively quickly. Certainly in the day of a production in a milk processing facility, that's not too long. They develop most rapidly in a flowing and wet system where there are adequate nutrients and water and moisture available. So, you know, the milk lines certainly a perfect breeding ground for bacteria and for the development of a biofilm. Numerous species of bacteria can form biofilms, including some nasty ones like Pseudomonas and Listeria. Obviously, those microorganisms in particular can pose some severe health issues if they are to contaminate our milk or our food products. Non-biofilm forming bacteria, molds, and viruses, all different sorts of microorganisms can become encapsulated in that biofilm. So in other words, they don't need to be able to build the city or the structure in order to live there. Just like all kinds of proteins and food items and different nutrients get stuck in that biofilm, so too can things like yeast and molds and viruses. So it's a very diverse ecosystem. Microorganisms in the biofilm, because of the structure, the polysaccharide matrix, they're much more protected from cleaners and disinfectants than freely dispersed microorganisms, and that becomes a big challenge in fighting microorganisms that are encapsulated in that biofilm. Biofilms can be seen with the human eye if they become large enough. They can grow to 1 or 2 inches thick. But many of them are invisible without the aid of a microscope. Again, which makes it difficult to fight, because it's difficult to fight what you can't see, what you don't know is there. The formation of biofilms. Bacteria can attach themselves to surfaces such as metal and glass and plastic by weak polar forces that are similar to static electricity. Many bacteria also have the capability of attaching themselves to a surface by means of finger-like structures if the conditions are favorable for bacterial growth. In other words, if there's moisture and nutrients in an area, a bacteria might decide to attach itself to a surface. And once they are attached to a surface and they are capable of forming a biofilm, they begin secreting that sticky gel-like substance, that polysaccharide, that holds the biofilm together, and that polysaccharide gel acts as the adhesive, captures nutrients, and holds bacterial cells in close proximity to one another. Bacteria also actually communicate with one another through different chemical compounds. AHLs, or N-acetylated L-homoserine lactones, are one of those chemical triggers that once a bacteria starts releasing that, the biofilm starts to develop. It causes that hormone causes changes in gene expression, which results in the formation of pili for bacterial conjugation. So they stick together rather than flagella or things that help them move around in an environment. So that once they find a place that they like, they start sticking there. And this is one of the triggers that forms a biofilm. In the early stages, the biofilm, obviously, as it starts growing, starts out small, one bacterial cell layer attached to the surface. As the cells grow and multiply, the biofilm becomes larger and thicker and more layers. And again, they can range from very thin to where they're not visible to the human eye, up to several inches thick. During multiplication and growth, bacteria capture those nutrients, proteins, minerals, everything they need to survive from the surrounding area. And again, it's encapsulated and flown through that biofilm matrix. Some further characteristics of a biofilm. Here on the right, you can see that there are channels in which water and nutrients or milk, if it's in a milk line, can flow. So it's not just a solid mass. There is a little bit of a structure to it. There are areas that allow for flowing of oxygen and nutrients and so forth. So also the excretion of waste. So that biofilm matrix produced by the microorganisms help protect them from cleaners and antimicrobial agents as well. So within the biofilm, there are what you can call microenvironments, where dissolved oxygen and nutrient concentrations differ. So oxygen-starved areas in the biofilm, which are typically deeper, may yield slower or halted growth. Those areas are, again, typically deeper where the diffusion of oxygen and nutrients is not great. Nearer the surface or nearer the channels where you've got flowing oxygen and liquids, the aerobic respiration predominates, and you see typically increased growth in those areas. The biofilm has, again, some ways in which it regulates itself. So there are mechanisms in order to prevent those channels from closing up and preventing the biofilm from cutting itself off from the nutrients and oxygen and so forth that it needs as it grows. There are two types of microorganisms in a biofilm based on their position in relation to the biofilm. Planktonic are floating on the surface of the biofilm or near it, and those are relatively easy to kill. They're not inside the structure where they're protected. And then there's the sessile, which are embedded in the matrix itself, and they're anchored to the surface by those polysaccharides. And inside the matrix of the biofilm, it's very difficult to get down in there with chemical agents to fully inactivate all those bacteria, viruses, or whatever else may be encapsulated in that biofilm matrix. So controlling microorganisms that are just on the surface of the biofilm or around there are relatively easy to control. But controlling those doesn't necessarily mean that you're controlling the biofilm. In other words, you can kill the microorganisms living on the surface of the biofilm, but that doesn't indicate that the microorganisms underneath or within the matrix are inactivated. In other words, you could potentially take a swab on the top of a biofilm after you treated it with a disinfectant and find no growth, but there is obviously a plethora of growth underneath the surface of the biofilm. Again, a biofilm gives the bacteria and other microorganisms that are inside the biofilm higher resistance to chemicals. They're more resistant to cleaning and disinfecting. And part of that resistance is just because the compounds need to penetrate deep into the biofilm to eradicate all the cells. Much of the active ingredients also are kind of consumed by reacting with the matrix of the biofilm near the surface. So you may kill, surface microbial agents or surface bacteria near the surface of the biofilm, but it's difficult to reach down all the way to completely eradicate that biofilm. One of the reasons biofilms are so detrimental to the food industry and the milk industry is because they don't just stay inside the surface. When they're exposed to outside surfaces or outside forces or at certain points in the life cycle, they just kind of break apart. They spread in several different ways. The illustration below shows a couple different ways how they can spread. You've got things like seeding. Parts of the biofilm can break off due to flowing liquids over the biofilm. Water, milk, or other beverages inside of pipelines, for example, can cause those things to happen. Here's another illustration of some of the ways in which biofilms spread bacteria and other viruses, molds, and so forth, yeast. You can have streaming, detaching of the biofilm, seeding dispersal, rippling effect, and rolling, all due to the velocity and pressure from flowing liquids. So biofilm mitigation strategy. How do we control a biofilm? Well, one of the best ways to control it is to try to prevent its formation in the first place. So properly cleaning and disinfecting our environmental surfaces, the bacteria are much less likely to attach themselves and begin producing a biofilm to begin with. How do we do that is by getting back to the basics of plant hygiene. That's one of the best defenses against the biofilm formation. Some of the things that are key to that are a good first rinse, a dry pickup if we're cleaning the general environment, 100% coverage of cleaning compounds. A cleaning compound that doesn't contact the surface can't clean it, obviously. If we're doing a CIP process, clean in place process, the velocity, we want to achieve a minimum velocity, so we achieve turbulent flow, which provides a little bit of physical action that can help break up soils. The correct chemical concentration, correct contact time, and temperature are all critical as well in terms of plant hygiene. If we're applying sanitizers or disinfectants, we want to make sure we, again, cover the 100% of the surfaces with those antimicrobial agents. Oxidizing chemistries are good in terms of combating biofilms. We want to also use, at least on a rotational basis, acid cleaners or disinfectants that can help break down things like mineral deposits. Chlorinated alkaline detergents and disinfectants are a good option for mitigation because they hydrolyze proteins and neutralize organic acids within the biofilm. Active alkalinity as sodium hydroxide of at least 0.5% also helps break down the polysaccharide matrix of that biofilm. And depending on how large it is, we may need more than that. But 0.5 is about the minimum that we've found to be effective against the prevention of biofilm growth. We also may want to increase CIP circulation time with oxidizing cleaners and antimicrobial agents. Strong oxidizing agents help penetrate and remove that polysaccharide matrix, and they include things like sodium hypochlorite, which provide active chlorine, chlorine dioxide, peroxyacetic acid, hydrogen peroxide along those lines, and generally peracetic acids also contain hydrogen peroxide. And then there's also enzymatic detergents that are designed to enzymatically break down the biofilm, particularly the polysaccharide matrix. So here's just a quick illustration of sodium hydroxide-based products versus enzyme products. So down below, the two small molecules there is a representative of sodium hydroxide. And that, when it reacts with certain soils, becomes consumed by that reaction. So the hydroxide molecule binds to parts of the soil as it breaks it apart. So if we've got 0.5% sodium hydroxide, for example, at the end of a CIP cleaning, we would expect to have a lower level than that if we're titrating for active alkalinity. Conversely, an enzyme breaks apart soils and so forth to help catalyze the breakdown of the soils, but they're not consumed in the reaction. So a relatively small concentration of an enzyme can clean a larger amount of soils than a conventional cleaner like sodium hydroxide, because the enzyme is not itself consumed in the reaction. We discussed briefly that the rotational use of acid cleaners and antimicrobial agents is beneficial, and that is because that helps remove hard water deposits. If we've got hard water deposits on our equipment, that can actually stimulate the growth of a biofilm by providing an initial structure on which they can stick and grow. So keeping our environmental surfaces free of hard water buildup by using acids on a rotational basis at least is going to further help prevent biofilm formation. Even if our plant hygiene is perfect though, biofilms are still likely to form during the production day as soils and microbes that may be present in the milk or the water or whatever foods we're processing are deposited on equipment. And once a biofilm is formed, special cleaning may be needed to remove it. We do have some tools. There are some tools out there that aid in the identification of biofilms or residual soils on surfaces. These identification tools can be used to help determine if biofilm removal efforts have been successfully implemented. So if we know we've got a biofilm after our cleaning regimen and disinfecting regimen, we can use some of these tools and they will help identify if those cleanings and disinfectings have been effective at the removal of the biofilm. Here we'll just go through some real world examples of a biofilm. Common locations are rough welds, wet floors, drains, just any area where we've got standing water. A biofilm formation is likely. So drains are a big place for the formation of biofilms. Here we've got some biofilm slime at the bottom of some water holding tanks. You can see that it's pretty disgusting, and that's a huge problem if this is processed water, and we've got a significant biofilm growth. That's a big contamination issue. Undersides of processing equipment, operation hoses, electrical wires, and so forth. Anywhere we've got, you know, moisture in an area that we don't clean on a frequent basis, we can have a bacterial or biofilm growth. Cooling units and drip pans are another big place. We've got moisture. A lot of the problems with that is that they're overhead. What's up generally has a tendency to come down. So if we've got bacterial growth above, that can very easily spread to the rest of the facility. The foaming and sanitizing equipment that we use can also grow and spread biofilms and bacteria. You know, you've got the hose there, that's drug across the floors, it's drug across drains, all sorts of things. Drains can become backed up during the plant hygiene process. And then, you know, it's, unless you're paying attention to it, you don't necessarily clean your cleaning equipment. So in that instance, a cleaning hose or a sanitizing hose can become a potential point of contamination if we've got bacterial growth or biofilm growth on those. Some additional places are electrical boxes. You know, they're sealed during the plant hygiene. We've got high moisture concentration. You can get bacterial growth inside those. Buttons, control panels, touch points where we've got, you know, a team member with a dirty hand or a dirty glove touching that point. Again, we have to pay attention to any touch point there. Just difficult to clean areas that are not cleaned on a regular basis. You can have bacterial growth that can turn into a biofilm. So rotational cleaning of ancillary areas is very necessary. Common locations of biofilm also are maintenance tools. Dirty, unclean maintenance tools, you know, they're brought out onto the facility from the maintenance shop. In the maintenance shop, it's warm, perfect breeding ground for bacteria and other microorganisms. So because of that, in order to help prevent spreading of bacteria, we should have a cleaning and disinfecting regimen before we bring them out onto the processing facility or use them on food contact or beverage contact equipment. And then after we make a repair on a piece of equipment, we want to make sure that that is cleaned and re-disinfected. Water lines also can be a source of biofilm or a good place for biofilm to grow. You know, generally we've got treated water, but if that water sits for an extended period of time in a dead end leg, you know, that antimicrobial agent can become consumed, and then that allows for the growth of bacteria. You know, you've got moisture and potentially nutrients. It's not uncommon in a water line that you'll see a biofilm. Here we've got another couple pictures. Here we've got on the left a pretty gross biofilm in an exhaust fan. So that is blowing air potentially out onto our production facility. It's taking that bacteria and bringing it out into the environment of the production floor. Trench drains or any drains are a potential area. The third there, we've got a biofilm at the bottom of a product holding tank. And even in our CIP system, we can have biofilms growing. That's all we've got for now.
Thank you so much, Justin. That was very interesting, and I really enjoyed the very practical examples that you showed and that you spoke about. So to the audience currently, I see that it seems like you don't have any questions. It seems like Justin has answered most of your questions. But if not, if you can please take note of Justin's contact details and also Jeff's contact details that's on the screen. Should you like to contact them directly and ask your questions if you feel there's no questions? Justin, from my side, it seems that the most important part is that you need to try to prevent biofilms from forming. And the way that you've explained it, it's all to do with the way that you're doing cleaning and your chemicals and everything that you're using. But should you determine that you've got biofilms and the mitigation factors that you've mentioned now, how long does it take before you can actually be sure that the biofilms have been removed?
Well, sure is a strong word. It's difficult to know that a biofilm was completely eradicated. Again, you can have false positives because the biofilm is protected. You can, as I mentioned briefly, do some microbial swabbings directly over the surface of the biofilm. If, the surface microbes have been eliminated, you can get a false negative. And so it's difficult to know that you've completely eradicated a biofilm. But again, we do have some tools. I would say a general CIP process, at very minimum, 30 minutes. But if you know you've got a biofilm, extending that up to 2 hours with a high concentration of disinfectant is not out of the ordinary. And you have to take it on a case-by-case basis. And again, after cleaning and disinfecting, using some of the verification tools that are available is very helpful in helping determine if your cleaning and disinfecting steps have been effective.
Yes, and I think also while you were mentioning the mitigation factors is also understanding that why it's so important to understand what you are using to clean and how. So, when you were speaking about the CIP, making sure the pressure is correct and the contact time and temperature of the water and everything. So that just shows that your basic PRPs in terms of cleaning, training the cleaners properly and all of that plays also an important role. Okay, so I've got a question here for you. And I know in the beginning you said that you're not a microbiologist, but I'm sure of it, you will be able to guide this. Don't the viruses in the matrix attack the bacteria?
Not necessarily. Viruses are relatively specific in the type of organisms that they attack. So a bacteriophage would certainly attack a bacteria. However, if you've got general plant environment, a team member who's ill with a virus sneezes and that virus infected mucus lands on a surface, that type of virus can become encapsulated in the biofilm. Viruses are relatively specific in terms of the species that they infect, so it depends on what type of virus is encapsulated, but certainly a bacteriophage will attack bacteria.
Yes, yeah. Okay, then there's another question. When they built a biofilm, did they build a multi-species biofilm? So I'm not too sure if that question is clear to you. If not, I can ask Tanya just to elaborate, but does it make sense to you what the question is, Justin?
No, I'm not exactly sure what's being asked there.
Okay, so...
Sorry, I think Justin referred to recreating biofilm in the laboratory.
Yes, that's right. That's right. Thanks, Jeff. So yeah, so when they built a biofilm, so meaning when you built the biofilm in the lab, did you build a multi-species biofilm?
Generally not, no. Generally, when we do experimentation on biofilm, it's done in a biofilm reactor, and we generally focus on one specific bacterial species, biofilm.
Okay, so I assume by asking that question, biofilm can then obviously be built up by multiple species and not just then the ones like you've replicated it in the laboratory.
Yeah, it could be. Yeah, you certainly could. You would just have to introduce, into your biofilm reactor several different species.
Okay. Okay. Thanks, Tanya. I see you say that answers your questions. Okay, my next question is from Tabang, and Tabang is saying, what are the possible negative effects from the mitigation strategy? Because solutions tend to fix a problem and also pose other problems somewhere, somehow. So, Tabang, I am not too sure exactly what you mean by that. I see you've got another question here. You said, beside possible human diseases as a result of viruses and bacteria found in biofilms, do biofilms cause other problems in the location they are found? For example, being found in a dirty electrical box, can that cause harm in any way? Perhaps lead to the broken box. Okay, so I assume what Tabang's meaning is the examples that you used from where you said biofilms can be formed on an electrical box. Is that really a problem?
Yes, anywhere you have bacterial growth, there's the potential to spread to other areas. You could have bacteria and so forth that become airborne, that could infect team members. If you've got things in an electrical box, it could drip out onto the floor, very easily spread to other areas. If a team member opens up an electrical box and comes into contact with that on their hands or their gloves, that can be spread throughout the floor, the production facility. So yes, additionally, if you've got microorganisms growing in things like a cooling tower and so forth, that can greatly reduce the effectiveness of the cooling tower. So yeah, they can cause all kinds of problems.
Yeah, and also cross-contamination from people touching and then not washing hands properly, and that. Here's a very interesting question as well. Is the presence of a biofilm possible if not visible? Or is it fair to assume to say that it will be visible on a surface?
No, biofilms can be invisible to the human eye. You can have, microbes are very small, and unless a biofilm gets very large, they're predominantly invisible to the naked eye.
Yes, yeah, definitely. Tanya, one of the delegates in the room, said that, corrosion of pipelines, Tabang, can also cause the biofilms, and the corrosion of the pipelines itself create the biofilms. I think what's very important is the basic steps that you've mentioned, that when people, want to go through these systems, to understand that any area that not necessarily is visible to the eye but needs to be cleaned is areas where you may be concerned that there's biofilms. Like you said, the drip trays and behind pipes and where water is accumulating and all of that. But it's definitely sure that you don't want to make sure that you get biofilms, because to get rid of it seems like a very big challenge. Jeff, is there any other comments from your side, or Andrew from your side?
I think one of the things that Justin referred to was that through the process of prevention, best practice plays a large role in preventing biofilm, and that's your first base of protecting your food processing environment from the creation of biofilm.
Yes. Yeah, and sorry, just to explain myself better, what Tanya mentioned, she said that the biofilms create corrosion of the pipes. If I didn't explain it correctly or answered you correctly, Tabang. So it's definitely something that you want to make sure that you don't get into your pipeline.
It's an important question, what is the mitigation strategies? One of the big negative effects that you get if you start getting biofilm problems, to make sure that you get rid of it, is increased cost in your cleaning and increased production downtime. To make sure, listen, we've started getting problems with biofilm, we're trying to remove that problem. So to ensure that your quality of your products and things don't deteriorate quickly. So the best strategy and the cheapest strategy is to make sure that you, in every time that cleaning is done, it's done properly to prevent those bacteria starting to express the genes that make them want to form biofilms. So that because it's so hard to make sure, listen, we had a problem with biofilms, and is it gone 100%? Because as soon as it starts being expressed in those bacterial populations, it comes back easier. So the biggest cost of the biggest problem or the negative effect of these mitigating strategies is you have to do these mitigating strategies, because you don't want to do mitigating strategies. You want to produce product in a factory, but you have to do these mitigating strategies to make sure that you don't have bigger losses.
Absolutely, Andrew. I think you hit the nail on the head there. And I always, from my days when I was doing audits and things like that, it's very amazing to see sometimes the cleaners or the people that are being utilized as cleaners within facilities are sometimes people that are not educationally very strong, or they've been with the plant for very long, but many a times the training of those people, it's just quickly run over because it's just a cleaner, but if you take it that person is having a very important function within the factory. So it's important to ensure that you give the correct attention to training, especially when it comes to the chemicals. And many a times you would see people, there's a measuring cup for the chemical, but no, it's okay, I'm just going to measure it with the eye, and you go gluck, gluck, gluck into the container. But these are all important factors that help to ensure that you don't sit with these type of problems, like creating biofilms in your factory, and in your pipelines. I think, Jeff, any closing words from your side?
Once again, I would like to thank everybody for showing an interest in this subject matter. And a very big thank you to Justin for sacrificing that beauty sleep all the way from America to attend and to present. It was, for me, very informative, and I trust and hope it was the same for everybody else.
Yeah, I do too. One other quick thing also on a potential negative effect of biofilm mitigation is the increased use of disinfectants can lead downstream to negative effects on the wastewater microbial populations, and that again can be an expensive venture. If you're killing off all your wastewater bugs, that's not a good situation to be in either.
Definitely, Justin. I think, like someone else mentioned, if you mitigate on one side, what is the effect further down? But I'm sure of it, you guys will all be available to advise any of the delegates should they have any further questions. But at this point, I want to say thank you to the Dairy Standard Agency once again for this webinar and hosting it with us. Like all the previous webinars, this webinar is recorded, and the presentation and the recording will be available on our website, www.foodfocus.co.za. So you will be able to share it, you'll be able to go and listen to it again, should you feel that you've maybe missed important points. And we are also looking forward to another webinar that will be hosted on the 30th of August, also from the Dairy Standard Agency. Please have a look out for the topic and the registration. We will most probably start to promote that for the end of the week. Other than that, I want to say thank you to everybody. I want to say please take a few minutes after this webinar. There's a short survey. If you could please answer these questions, because that will help us to make sure that we stay on point with the type of topics that we present to you. But other than that, I want to wish everybody a blessed Tuesday and yes, the rest of the week. And thank you very much. Goodbye.
Thank you.